Everyone, welcome. My name's Austin Davenport, if you haven't heard yet. I am a cultivation coach here in the Central Texas area, and I make it my mission to meet you wherever you are and help you grow towards your goals. I'm a generalist by trade, so I'm always looking to have as much learning experience in all the different things that I can get my grubby little hands on. And today we're going to be learning a little bit about applied microbiology in terms of using trichoderma fungi to boost plant processes for purposes of horticulture, agriculture, all that sort of nonsense. It's going to get a little complicated, but I've done my best to kind of generalize things and make it as understandable as possible. I'm going to be doing a lot of sweeping generalizations. I'm not going to be using a lot of words like rhizosphere, even though that's really important. So let's just get started. Today, we're going to be learning about a domestic shift happening in agriculture as people move away from harmful pesticides, fungicides, herbicides, and heavy fertilizers towards things that are a little bit more soft-handed. And then we're going to get into like what is trichoderma. There's a lot to get in there. We're going to go into the applications for trichoderma, how it's being used in agriculture. We're going to cover how to apply it yourself, and we're going to learn how to propagate it. So let's get right into it. But first, as Angela said, we're CTMS. Our main pillars are education, citizen science, and our block program. You can get connected with us through our many cultivation workshops. We do walks. We do a monthly guest speaker series that you're participating in now. If you go into our backlog on our YouTube, you will find dozens of really interesting talks on things like using mushrooms for textile dyeing, mushroom photography in the wild, getting into bioremediation, and so many really amazing things. I highly encourage you to deep dive into our past live streams. We also have our amazing citizen science branch where we are looking to help new and budding um, former parks that used to be former landfills and things like that to try to encourage tree development using mycorrhizal inoculants. And we also have a wonderful assistance program you can get involved with at the Fungarium at UT, helping to identify things, and you just really get to learn a lot and hang out in a really cool space. And then we have our flagship program, our mushroom block giveaway program, where we look to reduce waste streams that come out of the mushroom farms and increase local food access and promote soil health. Uh, Real quick, I do like to keep my talks fairly conversational. If you got a question, if you got a comment, feel free to like use the Zoom ability to raise your hand, or you can fire it off in the comments, and hopefully Angel can catch me as I'm deep into thought or whatever. And I also give away my presentations at the end of every one of my talks. Anytime you come to one of my presentations, there's a QR code at the end. Take stuff with you. I don't like people doing the work twice, so I highly encourage you to grab a copy of the presentation. As we move along here, I wanted to highlight a little bit more about our block program. As we move into the warmer months, these mushroom blocks can get easily contaminated by trichoderma fungi. And this is trichoderma fungi that wants to either inoculate and attack the mushroom mycelium itself or start to colonize the substrate. And that really can ruin processes revolving around, I want to grow mushrooms off of a block. But if trichoderma is there, it for the most part will like outcompete and start to overrun the block and become a little bit more of a health hazard than something that will produce food. But that doesn't mean that that block is useless. In fact, its amazing benefits move away from producing food towards producing healthy soil. And you can use these blocks in composting operations, smashing it into the base of a tree, smashing it into your garden, all that sort of stuff. And it'll really, really go a long ways to helping soil health. So this is just something like seasonal that goes on in Central Texas, specifically with our block program. In the cooler months, the blocks are amazing at using in cultivation practices, growing your own gourmet mushrooms. And in the warmer months, as these blocks get higher rates of contamination of trichoderma fungi that loves to be around in temperatures 80 and above, those blocks are way better off used in soil health and composting. I really quickly want to highlight our mushroom block page showing you a map of over 30 different locations where you can go and find these blocks. These are all static sites that have offered their space to us. And all of our hosts just have one condition. We only have one condition for our hosts. You tell us when the blocks are empty, and we go out and refresh those. So if you look on this map and you see all those green zones, those all have blocks. The red zones don't. So before you go check, before you go run out and grab blocks, check this map. And that way you don't waste your time. If you really, really want any, if you're looking for blocks, more, if you're looking for more than 50 blocks, reach out to us. We have a page for that. Contact us and we'll coordinate with you. You don't need to be bothering the mushroom farms. There's plenty of people bringing up their phones at all hours of the night trying to get these things. But if you go through us, we'll handle it and we'll kind of alleviate the mushroom farms from the excessive communications. Would there be an ill effect from breathing the trichoderma from the bags? Yes, Yvonne, I would definitely say the spores can pose a bit of a health risk. So anytime I'm using the blocks, and I have this mentioned a little bit later, that are heavily infected with trichoderma, I'll put on a mask. I still have plenty of mine KN95s from a few years past. They're could pretty much always be on hand for everybody, especially as we talked about that documentary earlier today. Um, when using trichoderma from these blocks, we want to be using a mask, pretty much hands down. No contentions there, no changes. Got to use a mask. Oh, so really quickly, there seems to be a perspective shift in general agriculture, definitely in regenerative agriculture, and that is we we are looking to like reduce the amount of heavy-handed pesticides, synthetic fertilizers, those sorts of things, and instead bring in what are called biologicals, biostimulants, biofertilizers, all these sorts of things that just involve using some sort of normal, run-of-the-mill organism that occurs in the wild, propagating that and then introducing that into spaces. You could think of things like humic and fulvic acids, biochar, liquid seaweed, microalgae, and trichoderma. There's plenty more to get into, but these are just some basic examples to get you familiar with the term biostimulants. Here in the United States, there are, okay. Yeah, about 30% of farmers globally are using biostimulants. I saw lots of records of trichoderma being used in East Asia, in Brazil, and plenty of other spaces. So we're just kind of like late to the trichoderma party, if I was if I was to take a guess. Um, but the emergence of biostimulants offer a more holistic, a more gentle alternative to pest and disease management that we find ourselves creating so many problems today. And this is Spence, otherwise known as Mama Microlife. And there's a little bit of a definition of biostimulants that I like to defer to Microlife for. Here at CTMS, we're big fans of Microlife. I use it all the time in my garden. They're not a sponsor, they're just cool people. And biostimulants, by their definition, catalytic agents that promote healthy growth, positive soil microorganisms. Those are things like molasses, humates, fish, seaweed, and organic fertilizers. So the idea is just keep it organic, keep it chill, nothing too heavy-handed. And in the copy of the presentation you can get, all of these little links are live, and we'll just keep on moving down the line. Oh, right. So there are many species of trichoderma, but there are very specific species that are used in agriculture that are typically root colonizing saprotrophic fungi that are so evolutionary successful that they are found in nearly all soils across the globe. This is a species of fungi that is so good at what it does that it's been able to propagate itself almost everywhere where there isn't a polar ice cap. There are so many variations, like we talked about with the mushroom blocks. Those are variations that love to colonize and live in either other mycelium or in the substrate of the block itself. What we're going to be talking about today, and what I'm mostly going to be focusing on, are the different types of trichoderma that love to infect plant roots. So there are trichoderma that live in mushroom substrates, that live in mycelium, and then there are ones that live directly in plant roots or even around plant roots. And most of the time where I am saying the word trichoderma, I could be talking about one specific strain. I could be talking about a grouping of strains, or I could just be talking about trichodermas on the whole potential. So it definitely varies. And all across the world, this stuff is being used as either a biofungicide, a biopesticide, a bioinoculant, a biostimulant, or a biofertilizer. And that just really gets into like, there's so many things that trichoderma is being used for out in science and in being applied out in the fields to do benefits for agriculture. All right. There are well over 250 strains of trichoderma, and there is no catch-all for general application. And it can quickly become an uphill battle trying to find the right strain for the right job. As we just saw, all of these different fields is either a fertilizer, a fungicide, a pesticide. These are, for the most part, different strains of trichoderma doing these different jobs. And it's really hard to find that catch-all one. And this becomes a key struggle in home scale propagation of the wild trichoderma that infects the blocks and stuff like that, because we don't know what strain that is. It might not be the beneficial one. It could be inconsequential. It could be pathogenic. We really don't know, but it's not too much of an issue. Yeah, I could show you how trichoderma looks like on a block. I actually have our, where's our trichoderma page? Da, 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 da. All right, real quick. Bam. So this is what trichoderma looks like growing on the blocks. You can see it on our main page about trichoderma. And we have a few photos here showing you exactly what that looks like. It's green. It looks nasty. It definitely looks like you shouldn't be inhaling it because you absolutely should not be. But any sort of discoloration on the mushroom blocks, whether it's oranges or yellows or these deep greens, that's kind of a sign of like, hey, this is good for composting, not so good for cultivating mushrooms on anymore. Thank you for your question. All right. Back to picking the right strain. If I am using mushroom blocks from the program that are infected with trichoderma, that's still a saprotrophic fungi. That's still speeding up the process of decomposition. So I am more than happy to use those by setting up a new composting amendment, whether it's a Johnson soup bioreactor, whether it's just a normal static compost pile, whatever it is. Infected blocks, blocks infected with trichoderma still have a great use in general composting operations. All right. Without explicit identification, it's anyone's guess as to what's going on here. Um, if you're a gardener, it's still worth using in a bit of a composting operation. But for general agriculture use, if you are a small scale regenerative farmer, you should be looking to get specific identified strains and apply those as they're intended to be. So you got to get the right tool for the right job. And anytime you're digging with trichoderma, you're going to be deep in the toolbox trying to find the right screwdriver for the right hole. Cool. So let's talk about one of these in specific, Trichoderma T22HC. Bioworks is a company that is doing a lot of good business, and they have a lot of good camaraderie the in agriculture as promoting and creating all of these biological amendments. The number one flagship product of their portfolio is called Root Shield Plus, which actually uses two different strains of trichoderma in tandem to help get all of these benefits here. Is there a strain that is beneficial to trees? There's a lot of research trying to figure that out of applying trichoderma, but and I was looking for resources to put in here about trichoderma and its potential as a treatment to oak wilt, but I didn't find anything concrete enough. Needless to say, Trish, it's just something that's ongoing. We'll breed it into existence at some point. The genetics of trichoderma are so strong and able to be like cultivated and bred and get all these new offshoots and new breeds and new cultivars of trichoderma. It's just such a prolific fungi that uh, these companies are able to, in a lab, breed the selected traits that they really want And so maybe eventually it looks really good, but I can't say that there's hard definitive evidence of a specific strain or a new strain coming out that is beneficial to oaks and oak health. But that's just what I found. I need to take a moment to remind you that I am not a microbiologist. I do not know the nitty-gritty details of all the things going on. Don't work at a university. I'm just some guy who likes creating PowerPoints and learning. But we'll go back to this guy, T22, the specific strain that is most common for agricultural uses. So people are using this as a seed coat, as a liquid injection into their soils, that sort of thing. And it does all sorts of stuff as like preventing fungal borne pathogens, creating root systems to become stronger, uh, increase moisture uptake, all this sort of stuff. And you can actually click on this gentleman here, Dr. Chris Hayes. I tried to find a better photo of this gentleman. I could not. I think that's a pretty serviceable photo. But this takes you right to their page about T22 and how they use it. I ripped all the bullet points for this. And they have a really cool compatibility tracker on their Bioworks site telling you, like, hey, I plan on using microlife uh, liquid AF treatments. Is that compatible with your products? And they went and checked like 400,000 different agriculture products to see which are compatible with their Root Kill Plus and which ones aren't. But I found that to be incredibly interesting and a great service to the greater community. But yes, and just as a reminder, as you get the PDF, all of these links are live. Everything that says click on me will work just for you. We're going to get on moving. Cool. So tomatoes, peppers, eggplants, cucumbers, strawberries, grapes all suffer from heavy bacterial and fungal disease issues. Things like powdery mildew, gray mold, dampening off, blight. These things are super common in our area because we live in a subtropical climate. It gets pretty humid here and can get intensely humid and intensely hot at the same time, which really just ramps stress and disease. Potato, beets, carrots, and onions are also susceptible to things like root rots and blight. So when you're monocropping these things, and this is what I was finding specifically in India, where they just continuously put the same crop in the same space over and over and over and over again, and you start to naturalize diseases for those crops exactly where you continuously are planting them. And what they would do to combat this is trichoderma treatments to prevent those diseases, keep the plant strong. And even though they were breeding those diseases and the exact things that wanted to those diseases wanted to infect, they were able to subvert a lot of that damage, a lot of that crop failure by using trichoderma treatments. Oh, right. Uh, Seemingly the strongest use case for trichoderma is as a biofungicide. It is so good at attacking other fungi in the soil that want to infect your plant, but it's not a wholly eradication. What they do is they, as Root Shield Plus would kind of allude to, they infect the plant roots and begin to create a gigantic field that just kind of waits for something to come by and punches it right out of the plant roots way. So those diseases are still fully present in the soil, but they're just not able to infect that individual plant that gets infected beneficially by trichoderma fungi. And it starts to release all of these little enzymes that will break down fungal cell walls. So anytime a predatory fungi comes around, trichoderma steps in and just kind of cuts it off at the base before that disease can propagate and begin to feed on your plants. Again, does not eliminate. It's a very soft thing. It's it's Bruce Lee. It's just like palm deflecting bad guys from your plants. It's not a whole cure, ripping out pathogens from the soil. All right. Fruit trees, apples, citrus, stone fruits, pomegranates, all these things that people really like to grow here can become susceptible to lots of different fungal issues, scab, canker, brown rots, and this one that I can't pronounce that I listed here. And if you typically think about like how we set up a fruit tree orchard, we are putting trees pretty close together because we want as much cross pollination as possible. If we are putting things in such close proximity to each other, we're also starting to encourage disease. And if we are monocropping things, if we just have one tree of all the same very close-knit genetics, we are just further inviting disease to come in and ruin things. If you think about the banana industry, if you think about um, citrus in Florida, what we did is we created these country law states or sometimes country wide monocroppings of one specific species. And it just takes one little bad guy to come in there and just wipe out an entire industry. All right, Gwen, if we make a trichoderma tea, do we need to dilute it prior to using it to water our plants? Yes. And any sort of trichoderma-based product you do find will have some sort of instructions on the packaging. Dilute 2 teaspoons per gallon of water. Maybe it's in liters if you're going across seas for products. But any sort of trichoderma thing you do buy will have instructions as to how to dilute. Oh, right. Um, So treating our fruit trees with a diluted trichoderma application will go a long ways into controlling a lot of these issues. Cotton root rot is also a huge issue for a lot of cultivators I find around town, especially ones trying to create a small orchard environment. If you are trying to create a fruit orchard, you are going to be inviting in quite a bit of disease, and we need to be very careful as to how we control that. A great way to do that is to just be as preventative as possible. Stay on top of your pruning, stay on top of um, keeping root health correct. Maybe infect those roots with some trichoderma to overhaul their resilience to pathogenic fungi. All right. I mentioned a gentleman here called Gary Harmon, and he has been studying trichoderma and its positive benefits since about 1980. He's been deep in the weeds of trying to find exactly what's going on under the hood on a molecular level, what is happening on the plant root zone, and how things are shaking out in a lab setting. And I have here 2 trichoderma species or 2 trichoderma strains that have both been separately shown to have significant effects on plant growth of annual crops. And what happens is the trichoderma comes in and it's like, hey, I have some things that you could use. You have some carbon I could use. Let's make a trade. And that way you don't have to have these processes done by yourself. I'll help you out. And I live in the plant roots, so I'm intrinsically invested in your health. So these trichoderma species I have here, Azarum and Varens, these things need to live on plant roots. And so they are super invested in making sure whatever plant that they infect lives, thrives. So they do things like boosting root structures, putting out all those enzymes that um, react with pathogenic fungi cells and reduces their presence. And then they will also increase photosynthetic potential. So they just, for some reason, can make the plant just do better at whatever it is. And I'm not going to get into exactly how that happens. You can definitely listen to some of the resources and podcasts and read about things that I will have further on in my resource section. Um, but trichoderma also like will secrete acids to dissolve minerals. I talk so much about plants being really bad at uptaking nutrition alone. They need some sort of mama bird in the soil. They need good soil microbiology. We need a good community of protozoa, fungi, bacteria, and everything in the right balance and beneficials as opposed to pathogens, all doing the work to soften up that food for the baby bird, the plant roots. Trichoderma has been known to do that. And at the same time, its strong colonization abilities will actually stretch out the connections between the end of a plant root and the subsoil that the plant root is not currently present in. And it'll give access to the plant to that subsurface soil so that it can take control and use some of those resources, those nutrients that are deeper in the soil than the plant roots are currently present in. It's really interesting stuff. All right. Fungal diseases and Texas. This is kind of what got me down the trichoderma rabbit hole to begin with. Um, Last year, I had my entire community garden bed just laid out with about 40 plants of about 10 different heirloom varieties. I fertilized correctly. I pruned vigorously. I love pruning my tomatoes. I took really good care of them because I wanted huge results to share with the community. and a blight issue or a powdery mildew issue just wiped out the entire bed. But that's kind of on me. Growing tomatoes specifically in a community garden context is really a big gambit because if someone's not pruning well, if someone's not taking care of their tomatoes, and I'm in very close proximity to them, that disease can just like pop to the next plot, to the next plot, and then it eventually find me. So I was very curious. I was like, I thought my fertility was good. I thought my practices were good. I thought my plants were in good health. What the heck happened? And earlier this year in January, I got to sit at a talk at the TOFGA conference, Texas Organic Farm and Garden Association conference. And one of the presenters was Daniel Lescavar, who is the lead tomato breeder at Texas A&M Uvalde. And what they're doing over there is they're trying to study how they could make tomatoes more heat tolerant and disease resistant. It used to be when you went to HEB and got a can of tomatoes, that those tomatoes were grown here in Texas. There used to be a booming Texas tomato industry. But as our climate has shifted and as things have gotten hotter, diseases have come along faster, that whole industry is just non-existent. And they are wanting to see what they can do to bring that back to life. And the two main things that they found were that created more success and more sturdy tomatoes was one, trying to breed new disease-resistant tomatoes. So they were out in Chile trying to find really crazy distant cousins of nightshades that are heavily heat resistant and that they could breed with tomatoes to create new tomato strains that hopefully are also disease resistant. And I actually got some of their seeds and I have them growing out in my yard here, and we'll see what happens. I can't tell you how I expect that to go, so it's just anyone's guess. But the second thing that they found to be incredibly useful was grafting. So if you take a hardy rootstock of a tomato and then transplant and graft it on your end plant that you really want to grow, the disease resistance was just through the roof. And I was thinking, well, if I can have a bit of a threefold issue, I don't really want to buy tomato rootstock. If you go on Johnny Seeds right now, their tomato rootstock seeds are like $45 for like 10 seeds or something. It is a big hit on the wallet getting tomato rootstock seeds. But I did ask him, I was like, hey man, can I just get some Matt's Wild Cherry, which is a cherry tomato, a typical cherry tomato hybrid mixed with a wild cherry tomato off of the mountains of Mexico. It was just bred together, and it's a really good tomato variety that gets you great production here in town. If you really want success with tomatoes, I would start with Matt's Wild Cherry. And I was, can I just use that as a rootstock instead of the 50 cent a seed alternative? And they were like, yeah, maybe. I'd give it a try. So I'm just trying to see what I can do to really like keep that total tomato collapse from happening again. And trichoderma and maybe grafting with some Matt's Wild Cherry will do it for me. But that's what got me really down this rabbit hole. Applications. So when you're trying to apply a trichoderma-based product, a trichoderma dominant product, typically what we do is we would coat our seeds prior to germination, either by putting them in a container with the dried trichoderma powders you get from these products, and just give it a good shake and let it rest for overnight, or you could let them soak in that liquid solution of trichoderma prior to germination for like 5 or 6 hours. There are a lot of seed coating products that are fungicides, pesticides, all sorts of things. The idea is we really want to catch that seed just as it shoots out that first root out of that seed. We want the trichoderma to catch that, infect it, and then begin that beneficial relationship right out the gate. And then the best practices, you typically would follow up with multiple rounds of it being diluted in water that you drench the soil with, and then come back in and do deep water to really take that trichoderma and put it back into the root zone. We always are going to be trying to introduce trichoderma straight into those roots. If your plant has a pretty short life cycle, if we're talking arugula, if we're talking lettuce, then you may only need to do a water application of trichoderma once. to see good results. But for longer season things or perennial crops, we're typically applying like every 2 months regularly, especially through like periods of stress. As we're moving into triple digit weather, I would be really, really on top of like making sure that my calendar has my trichoderma treatments set up. Um, I have I'm putting together a little bit of a experiment outside of my yard. I have one row of tomatoes followed by two close rows of zinnias, which also get exposed to massive amounts of fungal issues as we move into the hotter months. So I basically set the deck to have fungal problems in my yard, and I've been introducing trichoderma to that. And so far, so good. We'll see what happens. I actually had some brown rice that I had set up for another inoculation of oyster mushrooms ready to go, and that did get exposed to trichoderma. And then I was like, I'm gonna just pinch this into my tomato crop right here, and I could be doing myself a disservice. Again, I'm just shooting from the hip as to what the heck I'm propagating. That trichoderma that was infecting that rice could be a good guy, could be a bad guy, could be I don't know guy. I have no idea. I didn't look under a microscope. And it's kind of becoming a little clearer to me that it almost seems like for regenerative agriculture, for small scale agriculture, if you are using a lot of trichoderma based products, might actually want some sort of microscope. Um, you might want someone who's adversed in soil microbiology to be on the team. It's becoming more and more important as like these new biostimulants are coming around. We really want to be able to take a microscopic look under the hood and see how things are playing out in reality. There are backed up um, studies and recreations of successes with trichoderma in the labs, but trichoderma success replications in the field, not there yet. And that can be very disheartening to me personally because, like, hey, I want this stuff to work. I want to get some of this stuff. But if I'm paying $100 a pound, they better work. Oh, all right. So going back to using local wild trichoderma, if I'm using them in the blocks, if I'm taking advantage of whatever trichoderma is infecting that's just on hand that I have no idea what it is, I don't know how to identify that. It's still perfectly fine to use that stuff in composting operations. Um, these are some Johnson Sioux bioreactors we put together at Circle Acres that you can go check out on Saturday. I'm sure they're doing just fine. They've been heavily infected with slime mold and stuff like that in the recent months. But if you set one of these up with a bunch of trichoderma infected blocks, that's going to break down that all of that organic material even faster. Trichoderma, just like most other fungi, they're decomposers. They're here to break things down. And if you are going to use them, you need to be wearing a mask. And I'm just going to keep saying that because I want you to stay healthy. I don't want any unnecessary trips to the ER where you'll tell the guy, "Hey, I think I have some trichoderma in my lungs." All right. Yeah, no spore lung. We don't need that. Would trichoderma compete for nutrients in the soil? Not necessarily. It would outcompete a few things, but not anything that's really noticeable. And it's definitely not going to outcompete like mycorrhizal fungi. So trichoderma infects plant roots and has a beneficial relationship with plants, but trichoderma itself is not categorized as being mycorrhizal. So we really don't run any gambits of like harming anything that's also positive that could be competing in the soil. Yvonne, I wish I could talk more about it being about the pathogenic side of trichoderma. I just know that it's out there. There really aren't a lot of studies being like ringing the alarm bell of like, don't put trichoderma out there, don't do this or don't do that, or like there's this really awful strain. It's sort of just like agreed that there are pathogenic strains of trichoderma, but where those are, who those are, there's not a whole lot of talk or science on the matter, which makes me think that like it's not too risky to just shoot from the hip and apply wild trichoderma. Is common bread mold a type of trich? I have no idea. Yeah, yeah. Pathogenic Trichoderma searches on Google will definitely start to get a little scary, but I think searching anything a negative connotation on Google will try to reinforce that that negative or scary side of things. All right. So when it comes to sourcing trichoderma, it can be a little tricky. I've been trying to find like liquid cultures. I reached out to Bioworks. I was like, hey, our organization's looking to eventually see if we can set up some cultural libraries, much like the local seed libraries at the libraries have, where you can come into any library around Austin, grab seeds, check them out with a library card, and go home and use those seeds in your own garden. And those seeds are arguably the best seeds for our area. They've been acclimated to our area, and here at CTMS, we are super like keen on trying to figure out if we can crack the nut of like replicating that same kind of program, but using fungi. Like we want to give you agar plates of oyster mushrooms or lion's manes or all these sorts of things that you could use. On top of that, I would love it if we had some trichoderma T22, so those specific strains, not just propagating the wild stuff, but having those specific beneficial strains that we could give out to the community. And BOKASHI Earthworks actually does have great spore powders of T22. Let's see here. What did this cost? Again, it's a nice button, so you can click right on that to give you an idea of what's going on. All right, $34 for a vial. That looks delicious. All right, cool. There's the product page. We're giving away some of this stuff a little bit later on. There is, yes, it gives you the dilution rate here. And this is just straight T22. There's nothing else in this vial besides that. All right. And if we get genetics of trichoderma, we want to like set up our lab environment. We want to be as sterile as possible. It's really interesting to like think about traditional home mycology where you're trying to avoid contamination of trichoderma. But in this instance, we are trying to avoid trichoderma contaminating some trichoderma. And how that can go can vary wildly, but you always want to hedge your bets by creating a sterile lab environment. And I have some quick tips about that uh, right here. So to prepare your lab environment, we want to be working with a still air environment. We want a space that is either within in proximity of a blow hood. If you can't find one, they're like $100 to make DIY. Maybe the closest community college would be receptive to you being like, hey, I'm trying to do some transfers of fungi to agar plates. Can I use some of your space? Maybe you know somebody who's really into mycology and is currently growing a lot of things and transferring a lot of things, and you could try to use their space. You could take a a plastic tote, cut some holes in it for your hands, and do all of your transfers in that little space as if that was your lab, because that is a still air environment. You definitely want to kick off the AC. We don't really want things moving around in the air that could hit our agar plates. And then after that, we just want to sanitize everything. We want to sanitize our gloves. We want to sanitize our agar plates. And you can find pre-poured sterile agar for like $20 for 10 plates online. You could pour your own sterile agar. That's a whole other thing that I just think is too much time and effort. I'll let some lab deal with that and outsource that for $20. I would prepare my genetics depending on what state it's in, spores, liquid filters, etc. And then I would place that on the agar, again in a still sterile environment, sterile practices, that sort of thing. And then we can use some mycology tape to like seal the agar and put that in the fridge, ideally in a separate fridge than the rest of your goodies and food. But I personally keep my genetics in one of my bottom crisper drawers. I have a few different Tupperware containers, and that's where I'm storing my agar plates. And I make sure that they're labeled. Most mycelium looks the exact same. Trichoderma is going to be green, and trichoderma doesn't look any different than any other trichoderma. So we want to be careful with like labeling, putting a date on there. For the most part, fungal um, your fungi genetics that are colonizing agar plates will have about a 3 month shelf life, and then once that 3 months is up, you can grab those plates if you haven't used them yet. Just take a sample off of that back in your sterile lab environment and transfer it to a fresh agar. Sets the clock right back 3 months, and you're good to go. And it's just such a glaring like opportunity to create a cultural library. I'm just simply get obsessed with it. But that's just it's pretty easy. All you need is some 70% isopropyl alcohol that you can get at any place where there are nail care products, a clean plastic tote, and some gloves. And then you could be transferring things and doing mycology all over the place. All right. So we've talked a lot about trichoderma's use as a fungicide. It might have some benefits in treating oak wilts and promoting oaks. We've talked about it promoting photosynthetic potential. We've talked about a lot of beneficial things when it comes to trichoderma, but that's a lot of promise, and I start to get pretty skeptical once a specific pitch starts to get like a little too good to be true. And this seems to be a sentiment that's also shared by a lot of universities trying to replicate study results that have showed positive effects. This stuff is super, super complicated and has so many different variables around conditions, complicated chemistry, what's going on under the hood. Did you get like some contamination from a different trichoderma species that overrun the one that you're trying to use? These things are really hard to quantify and really get into like replicating positive results. I think my favorite um, my favorite uh, study on my favorite publication on this kind of concept is microbial inoculation of seeds to improve crop performance, issues, and opportunities. I believe this is the University of Oregon. It's one of the O's. I forget. They're all kind of like hanging out in the trichoderma world, all the Ohio, Oklahomas, and Oregons. But this is just a really good look at like a skeptical yet respectful view on like how trichoderma is playing out in trials and in the field. Um, there's about 200, I believe it's $200 million a year being spent on trichoderma based products for agriculture, and all those folks really want to make sure that that money is being well spent. And there's a lot of research into trying to figure that out, hammer out the details. How long does it take to see the results after applying trichoderma in the soil? It could be pretty noticeable within like 2 to 3 weeks of like, okay, my seedling, if I planted some seedlings on one side of my property and some seedlings on the other side of my property, if I seed coated these, if I gave them a trichoderma treatment at transplant, within 2 to 3 weeks after transplanting, I should see some significant difference between the ones I inoculated and the ones that I didn't. And I have a video later that I'm not going to show, but I have a link for it that goes really, really in depth as to like how things were developing as we had gigantic fields of celery with a control and a trichoderma treatment. But yeah, just want to say like if it's too good to be true, it's worth being skeptical about. Um, there's really positive things going on in the world of trichoderma, and there's also a lot of positive effects that are hard to replicate. And these are all some great videos slash podcasts that go into it. The first one is a um, a talk about using trichoderma in cannabis, especially in indoor areas. If you're farming cannabis indoors in a room, then you are kind of breeding you're setting the stage for a fungal issue to come in and just wipe everything out. And with cannabis farming, there is super amounts of there are impeccable amounts of scrutiny as to what products you can use, because these are things that people are ingesting in a lot of different ways, and we don't want to accidentally hit them with the wrong pesticide or herbicide that could harmfully impact your health. So there is a big amount of oversight as to what products can be used in cannabis when it comes to controlling pests and disease and stuff like that. And trichoderma has shown really good uses in those sorts of heavily um, secluded spaces where you are growing cannabis. And the yes, Dr. Chris Hayes from Bioworks is the guy being interviewed here. He worked at Cornell for about 20 years. He worked at Cornell for about 20 years, and he was studying all sorts of biologicals, bacillus and trichoderma and its uses. And then next I have In Search of Soil. Plants let trichoderma mold infect them. Why? Diego Footer, the podcaster here, has been podcasting about successful farms and small scale agriculture for about 12 years now. And I've been following him almost the entire time. I really enjoy the way he attacks things and tries to be, let's cut out the jargon, let's figure out what's going on in the play. And he actually interviews Dr. Gary Harmon, the guy that has been studying trichoderma since the 80s. So it's a really interesting conversation that'll robustly back up a lot of the things that I talked about here that I didn't want to get too complicated, too into the weeds about. Trichoderma fungi for forestry. This is rural delivery. These are, oh my God, I can't believe I can't remember right now if they were uh, from the UK or from New Zealand. Anyway, there's this is a smaller video that just goes straight into the details as to like how plant roots are being infected by trichoderma and what are its uses in forestry services. And then I think probably my favorite application of my favorite video covering trichoderma is this last one by Bioworks. This is where they are using about they go into 80 acres of they trial out trichoderma in controls in 80 acres of different mixed agricultural uses, for the most part in the Monterey area, the central California. And what they did is they started to infect celery, and you can see here over the years how things have developed as they started to reintroduce and reintroduce trichoderma inoculations into their fields. So these fields were getting wiped out by Fusarium, a fungal issue that'll attack a gigantic field of monocropped celery. And then they found over time using their trials that they were able to like really solidify strong health, resistance, photosynthetic potential, and the yield was almost 30% higher than the control group. They go into strawberries, they go into a few different things, and they have some graphs and charts all along the way with really impressive data showing like promising stuff. They did introduce the trichoderma in a liquid in the drip irrigation through a liquid dilution, but they applied a few other things here that I think really like make the trichoderma sing in on the stage. And this is product that you have to request privately from this company, Terra Ag and Technologies, and this is called Plant and Soil Pro 2. And what this does is kind of boosts the processes that trichoderma has on plants. So it's improving the exchanges and the transfer of beneficial nutrients into the plant. So fulvic and humic acid, specifically fulvic acid, is really good at transferring beneficial nutrients into the plant. So it's the delivery mechanism. It's what's getting that trichoderma right in that plant right off the bat and just kind of easing the whole process along. There's a bunch of other things in this product that has like organic sugars, things like molasses, some more complicated sugars to promote different kinds of soil biology, micronutrients, macronutrients, minerals, that sort of thing. And so they found that using a mixture of their Root Shield Plus, another product they had called organic, what do they call it? This is it. On Guard. Yeah. So they use their product On Guard, their product Root Shield Plus, and this mixture of humic fulvic acids, amino acids, and macro and micronutrients. And this is what created the three mixtures they put together did create significant results. And it's only a 58-minute video. This is a bit of a shot as to like here's the the Trichoderma trials, and here are the controls, and you can see that there's quite a bit more biomass on the Bioworks side than the control side. And I found this to be the most illuminating like window into how Trichoderma is being applied in agriculture. And these are all live links. So when you take the PDF home, you can just click on all these things directly, and it'll take you exactly where they lead to. I actually think we just did pretty good on time. There's our QR code. Uh, please feel free to continue to hit me with whatever questions. Trying to be respectful of your time.
Yes. Great information, Austin.
Thank you.
Yes, I see a few questions out there that we haven't addressed yet. So Chevy Chevy asks, how long does it take to see the results after application of the trichoderma in the soil?
After like multiple treatments, it should be like 2 to 3 weeks. You should see like a really good benefit as opposed to if you didn't give any benefit. But you wouldn't really be able to notice those differences unless you were like, this always gives me issues. If you're always having powdery mildew uh, with like a squash, and you plant it with a seed treatment, a seed coating of trichoderma, and you hit it again with a liquid drench of trichoderma on transplanting, maybe a month after that, you should start to see that big difference. But without having that control, it's hard to tell how fast that's really making a difference.
Mm-hmm. Yeah. So, and Yvonne asked, or actually, sorry, Marie asked, this was earlier on. You may have addressed it already, but she asked, can you make a fertilizer tea from spent blocks? What kind of fertilizer number would it create? And I've only seen soil samples of like completely dried up, like spent mushroom substrate. Like I've seen the sort of NPK numbers on that. So I don't know, but Austin, you might know more.
Yeah, the NPK values kind of vary wildly depending on like how much of the mycelium is still there, how much of the mushroom block did it break down, what's the available organic matter, nutrients. I don't know if I would dare try making a tea out of like infected mushroom blocks. All these companies, Bioworks included, are trying to get through the EPA a foliar spray application of trichoderma, but as we've talked about, that stuff's not good to inhale. I don't know if I would be brave enough to start
Mm-hmm.
spraying my plants with trichoderma, even if it was the second coming. I don't know, man. I don't really want to mess with that. I have decent health insurance. I don't have that good health insurance. I don't know if I would risk it.
Yeah. Um, just doing kind of some Googling too. Like there's like a couple of species that seem to be pretty typical that grow on mushroom blocks, but I like you were saying, like, it probably there probably hasn't been a whole lot of kind of microscopy, like looking at these species because there's a lot and the diversity is pretty incredible. Is and the source of the substrate, is really going to also maybe coming from another completely different bioregion. So even though like these species of trichoderma are soil borne, they're already here, it may have come from like a completely different part of the country if people are buying like pellets.
Mm,
Um, because like some of that stuff can live in like even like the processes, the industrial process of making wood pellets. Um, But I mean, just looking at some of the Googles, like there's a few species that are pretty typical that grow in the blocks that which are the, beneficial. And I do know like with a lot of spores, they're hydrophobic, which makes it easier to kind of, if you're concerned about breathing them in, making a tea will help maybe isolate it in some way, like where you can kind of they'll float on top of the water and be a little bit easier to like put on your plants.
mm.
Um, but yeah, I've definitely heard of a lot of people using the blocks just to make tea, not necessarily like take a contaminated block and use it to make tea. Um, but yeah, we'll continue to share information as we learn about it, as people do more experiments. And I have to say, like when I've kind of gone down the rabbit hole on this topic, I find a lot more content in Spanish
Yeah,
and in other languages, because a lot of, like, in Latin America and in Asia, the science is much further. Um,
it's
they're
so much cheaper. It's so much cheaper to
yes.
have like a trichoderma product than it is to have our synthetic fertilizers that are mined
Hmm.
way off in Eastern Europe, Eastern Europe that have to like track through all these spaces. So it is so much easier to like, okay, I got the culture of the right thing. I propagate it out. I sell it to my domestic markets, and that's why trichoderma is so popular in all these other countries because it is just a cheaper alternative.
Absolutely, yeah. And they're a little bit ahead of us in the kind of taking care of their soils. um, well, let's do the giveaway. So I did a random number generator, and I drew Clayton's name, Clayton Phelps. So, Clayton, I'll just need to get your address from you. But I think we maybe just met at the in Seguin. Is that right? You came to the class there. So yeah, I'll send you a message.
We had a couple new questions coming in. Can you test the soil? You test the mushroom block with a soil tester. That might be a question for Texas A&M. I garden in my apartments. There's a giant retaining wall right outside my back patio, and that soil was heavily compacted, heavily eroded, just absolute, almost concrete. I wouldn't call it clay soil. I would call it almost concrete. And over the last 3 years, as I've been living here, I've put about 300 mushroom blocks down, and it's turned into a beautiful layer of rich organic matter. I don't think the soil tests would want to test the organic matter layer that I've created, but they would rather see like 2 to 3 inches in the actual soil line and seeing what's going on over there. So I would definitely relay that question to someone who is a soil testing lab and see what their opinion is. What are the different methods of composting trike blocks? I would like to try to remove a few dozen blocks in soil. Um, I like to reduce the particle size as much as possible. So I have a cracked 5 gallon bucket, and instead of throwing that away, I kind of turn that into my block breaking bucket. And all I do is I throw one block in at a time. I take my shovel and I just kind of churn that thing like butter until it's an absolute small, fine granular, as small of a fine of a granular size as I possibly can. That'll just really speed up the decomposition process. Again, wear a mask. Got to wear a mask. Got to be safe. Michael said, I ran about 20 blocks that were infected through my wood chipper to break them down for my compost pile. Could just take a page out of Michael's book and grab a wood chipper for a much easier time. You guess that was a bad idea. I don't guess it was a bad idea unless you weren't wearing some PPE. Other than the PPE, you're totally fine. I've been using cement bags from commercial growers for a lot of different things, depending on the state of the material. Loose, minimally myceliated material, sometimes with trike. I've used it under our chicken roost to capture the manure, then compost. Any concerns you think I might have with respiratory issues? There definitely can be some respiratory issues when it comes to like digging out and cleaning chicken bedding. So if when in doubt, wear a mask. Just keep it safe. Can I vermicompost with spent substrate? Absolutely. And we've seen great local results, anecdotal. Nobody in CTMS has a big microscope practice. Maybe one or two of us does. But we haven't really looked at the vermicompost that was primarily fed with mushroom blocks. We've seen some really good stuff. Who was it? What was the vermicompost farm that
It was next to the
went?
mushroom farm. They moved to Kentucky,
Yeah,
and they were
Brothers.
called Brothers
Brothers.
Mushroom
Yeah,
Farm.
they were specifically grabbing all the mushroom blocks from a neighboring mushroom farm, and everybody I know that used that vermicompost was singing their praise. We all were a little sad to see Brothers Worm Farm go.
Yeah. And there's one study that I include in my talk that's about all the research on spent mushroom substrate and where they looked at spent mushroom substrate in worm farming. And there are really incredible benefits, especially with the like hormonal compounds. Um, so yeah, so I can put that talk in the link because all the YouTube recording has all of the studies in the links or in the description.
All right, I think we are ready to head on out.
Yeah. Thanks everyone for joining us. And just a heads up, next month we're going to be talking about leafcutter ants. So, Juju Dessert, who is doing her PhD, she's a PhD candidate at UT. She's studying these wonderful organisms, and we'll be talking about how they like to um, decompose different substrates, and she'll be talking about her study and just about their life cycle. It's going to be a very exciting talk. I saw it at Brackenridge Field Lab several months back for Science Under the Stars, and it's a very fun talk. So definitely encourage everyone to join us again online next month for Leafcutter Ant Talk.
Really fun. The evolutionary arms race of fungi and ants is such an interesting thing to get into.
Mm-hmm. All right, everyone. Well, thanks for joining us and have a good rest of your evening.